Welcome, everyone. The webinar is about to begin. Please note this call is being recorded. The Clinical and Laboratory Standards Institute is pleased to present today's online education program. This webinar is entitled Verification of Commercial Microbial Identification and Antimicrobial Susceptibility Testing Systems. My name is Alexandria, and we are very pleased to welcome all of you to this program. Today's presentation will last up to 60 minutes and will include a specific question and answer period. However, you can queue up a question for us at any time during today's presentation. You may submit a question by simply typing it into the Q&A box on your screen. Then be sure to click Send at the bottom. These instructions will be repeated later in the program. Our speakers today are Janet Hindler from the UCLA Health System, an active member of the AST subcommittee here at CLSI, and Beth Prowse from the Peninsula Regional Medical Center. Both speakers are involved in the production of the M52 document being spoken about today. Let's get started.
Hi everyone, this is Beth. I'm going to be starting our webinar today. Then I'll be turning it over to Janet. We hope that at the conclusion of this webinar that we will be able to describe a very practical strategy for the verification of AST systems. Also, we would like to be able to list the steps that are needed to verify the performance of new antimicrobial agents, including newly approved agents, when we add it to our existing AST system. And we also want to be able to explain to you how to prepare a verification report for AST systems so that you would be able to satisfy accreditation requirements. Most of what we're going to be talking about today comes from a new document from CLSI that was published last year. It is titled Verification of Commercial Microbial Identification and Antimicrobial Susceptibility Test Systems. This document is going to focus on instrument-based systems that we use for identification and susceptibility testing in the laboratory, but it can be used for other systems also. A couple of terms that we need to go over beforehand. The first term is verification. This is a study that's used to ensure performance when a new system is first introduced into a laboratory. Post-verification quality assurance is what we use to describe the ongoing processes, including quality control, that's used to make sure that a system continues to perform reliably as we use it in our labs. We use the term validation to describe what a manufacturer performs in order to set performance specifications. Validation is also the term that would be used when laboratories develop their own tests or when they use an FDA cleared test for an off-labeled use. I want to emphasize that the M52 is a guideline, not a standard. A guideline is a document usually developed through some type of a consensus process, and it describes the criteria for general operating practice. This is for voluntary use. You don't necessarily have to follow this guideline. And the end user can also use the guideline as it is written, or an end user can modify it to fit a specific need. A standard, on the other hand, like our M100 that we all use in our laboratories, that identifies specific requirements for a method or practice. It's also for voluntary use, but it should not be modified by the end user. So this M52 is a guideline, it's not a standard. Why do we have to verify our new tests in our laboratory? We need to do this to make sure that our system is performing within the manufacturer's specifications. We need to make sure that the system was installed correctly and it is functioning as it is intended. We also need to make sure that our laboratory staff can produce accurate and reproducible results in our laboratories. The staff has to make sure that they're following the manufacturer's instructions precisely for system handling, and they are performing the tests correctly. We also have to do this to fulfill regulatory requirements. When we are doing an FDA cleared test and we're using it as it is intended, we need to make sure that we are demonstrating performance specifications before we start doing patient testing. We need to make sure that we are verifying the accuracy, the precision, which is also known as reproducibility, and also, if it's applicable, the reportable range of the patient test results. If you are doing a test that is not FDA cleared, or if you're doing some type of modification of an FDA cleared test, before you start using this test for patient testing, not only do you need to demonstrate acceptable performance for accuracy and precision, but you also need to be able to demonstrate acceptable results for analytical sensitivity, analytical specificity, the reportable range of the patient test results. You need to establish reference ranges and any other characteristics that's required for test performance or for the interpretation of the results. Verification is required for any test system that is introduced into the laboratory for the first time. This is including when we need to measure an analyte that the laboratory has not previously measured. And it also includes a test that the laboratory currently performs on an alternative test system. Verification is also required for an analyte that's added to a test system, or if we do some type of modification to a test system. And this information is found in the CLIA Surveyor Guidelines, pages 175 to 176. We have a link there at the bottom of the slide. Anyone who has an interest in the quality practices for their laboratory, this is a good section of this document to take a look at, to refresh yourself with. Just an aside, verification is not required if you're just having a routine maintenance or repair done. If you're moving your system from one location to another location in the laboratory, the CLIA does not require a verification, but it does suggest that you do a day of QC testing immediately after the move and before you start releasing patient testing. When you look at the CLIA Surveyor Guidelines, the CLIA rules are very general about this verification process. It states that the verification of a method performance should provide evidence that the accuracy, precision, and reportable range of the procedure are adequate to meet the client's needs as determined by the laboratory director and the clinical consultant. A laboratory may use the manufacturer's performance specifications as a guideline, but it is responsible for verifying the manufacturer's analytical claims before initiating patient testing. What it does not specify is the number of specimens to test, the comparator method, and what the criteria for acceptability is. This has been a question a lot of people have had about IT, ID, and AST systems, which is why CLSI has come up with this M52 document to help give guidelines on what to do with these types of tests. Verification is what we're going to need to complete before we report any patient results. We will also be doing routine QC with each verification run. Just a reminder that by doing this routine QC during your verification process, it can help you eventually reduce the amount of QC that has to be performed. For the microbial identification systems, you can help you move to streamline QC. For your susceptibility test, it can help you move from daily to weekly QC. But as a reminder, you need an IQCP in place before patient testing begins in order to reduce this amount of QC that you are doing. We're going to first describe verification of microbial identification systems, so the IDs, on an FDA-cleared test. CLIA has performance specifications that it recommends for microbial identification systems. For accuracy, of course, it wants you to have an agreement between the comparative method ID and the test system. We would want to come up with the % probability or the confidence of that identification. Precision, are the identifications reproducible? Reportable range, the acceptable species IDs are provided in the database, and then reference range or normal values would not apply to microbial identification systems. You can find this chart in CLSI M52 on page 10. In M52, we go through what are the recommendations for accuracy and reproducibility, when to perform, how to perform this testing, the numbers and types of isolates that you should be doing, the source of these isolates. You know, we want to do patient isolates as well as some stock isolates. And how do you go about resolving discrepancies when you get two different identifications, when you do this type of comparison? We also go through multi-verification, how you approach that. CLSI M52, page 60, table C2, we have this chart here explaining some of the different organisms that you might want to take a look at when you're doing verification of your ID system. When you think about how many organisms you need to run for identification systems, you need to assess what type of change you are making. An example of a comprehensive type of verification would be needed when a lab had always sent microbiology to a reference lab, and now they've decided to start offering this service in-house. So they're bringing in a new microbial identification system, or they're switching from one manufacturer to a different manufacturer. Another example would be if they are converting from a conventional overnight identification panel to multi-top. Or if they decide to add a new panel for Haemophilus. These are all examples of a comprehensive verification would be needed. We recommend, or M52 recommends, a minimum of 30 isolates that would be necessary to be tested to verify accuracy. And to verify reproducibility, we would want to be taking 5 isolates. It could be 5 patient isolates, or it could be a combination of patient and QC isolates, and test them 3 different days. A limited verification would be performed if you're making if your panel has had a substrate formulation change. You would need to do a minimum of 10 isolates that would be affected by that substrate. And for reproducibility, you could do routine QC for an QC isolate affected by that substrate change 3 times in 1 day. If you're just doing a panel update, it's not necessary to run any isolates for accuracy, but you want to make sure that your quality control is performing correctly. A very detailed explanation of this is found in M52, and this chart can be found on page 18, table 3A. We're going to move on now to explaining how a manufacturer goes through the FDA clearance process for an AST device. The FDA and the Centers for Medicare and Medicaid Services, CMS, provide the regulatory requirements for bringing an AST device to market. So the rules for accredited clinical laboratories, we need to be able to use commercial FDA cleared test systems and we have to be able to follow manufacturer's instructions. We have to perform verification prior to the test implementation, and we have to perform extensive verification for any off-label use of this test. For manufacturers of AST systems, they must meet FDA performance specifications, so they have to receive FDA clearance. They must use breakpoints that are on the drug label, not CLSI, and they only report drug bug combinations with the clinical indication. CLSI also has recommendations for AST and reporting, but they are not regulatory. What does it mean for a commercial AST device to be FDA cleared? It means that the manufacturer has submitted 510K to the FDA. The manufacturer has also demonstrated that the results with their AST device are comparable to those generated from a CLSI reference MIC method. The FDA lists the data that must be submitted and defines the criteria for acceptability. We have a link here to that FDA document so you can see what that acceptability criteria is. And we also have a chart in the M52 in Appendix A. What data are required by the FDA for clearance of an AST device? There needs to be a minimum of 3 clinical laboratories that should be used. We want to highlight a couple of the data, particularly the organisms. We want to have a minimum of fresh, not frozen isolates, and we want to have also included in that previously tested stock isolates. There should be no more than 50% stock isolates. There's also a minimum of 75 challenge isolates. The challenge isolates are supplied by the manufacturer, and they should include some relevant resistant mechanisms that we're seeing in the clinical lab. The rest of this information is included in the reference that we gave you on the previous slide if you are interested in taking a look at this in greater depth. Next, we're going to take a look at the criteria a manufacturer uses to determine if the AST device results are acceptable. First criteria is the essential agreement. The essential agreement is defined as the MIC being within plus or minus 1 doubling dilution of the reference MIC. We would like to see the essential agreement to be at least 90% or greater. The category agreement is defined as when our susceptible or intermediate or resistant results agree between the test and the reference method. And we would also like to see the category agreement be at least 90% or greater. When a manufacturer submits their data to the FDA, one of the things that we're looking at are the types of errors between the reference method and the AST device. So the manufacturer is calculating very major, and minor errors that they have seen between the reference method and the AST device. Very major errors are defined when we had a resistant result with the reference method and a susceptible result with the AST device. The acceptable error rate for very major errors should be less than or equal to 1.5%. Very major errors are only isolates that are resistant by the reference method are including in calculating the very major error rate. Major errors are defined as being an isolate is susceptible by the reference method and it is resistant by the AST device. We should have less than 3% or less as the acceptable error rate for major errors. Minor errors are defined when a reference result is susceptible, but the AST device is intermediate, or the reference result is resistant and the AST device is intermediate. Also, if the reference is intermediate or the AST device is susceptible or resistant. These are all classified as minor errors, and there is not a rate of acceptability for these minor errors. That is not specified. When you calculate the essential agreement, we have the number of isolates that were within plus or minus 1/2-fold dilution of the reference method, and we put that over the total number of isolates tested and multiply it by 100. The category agreement is calculated by taking the number of isolates with the same SI or R result as the reference method and dividing that into the total number of isolates tested and multiplying it by 100. The error calculations. Very major errors are calculated by taking the number of isolates with a very major error and dividing that into the total number of resistant isolates tested, multiplying that by 100. Major errors, the number of isolates with major errors, dividing that into the total number of susceptible isolates tested, multiplying that times 100. And then minor errors are defined as the number of isolates with a minor error and dividing that by the total number of isolates tested and multiplying that by 100. On this slide, we have a hypothetical example of antibiotic A versus the Enterobacteriaceae, and we have the breakpoints listed. For isolate 1, we have a reference method where we got an MIC of 8, which is susceptible. For the AST test device, the MIC is 8. So it agrees, the essential agreement matches, the categorical agreement matches, so there is no error here. For isolate 2, the reference method gave an MIC of 8, which is susceptible. The AST test device gave an MIC of 2. Both are susceptible, so there is category agreement, but because the MICs are not within one doubling dilution, we do not have essential agreement. For isolate 3, we have an MIC of 16, which is an intermediate interpretation. The AST device has an MIC of 8, which is a susceptible result. There is not category agreement, but there is essential agreement since these are within one doubling dilution. This would also be a minor error since the reference method is intermediate and the AST device is susceptible. For isolate 4, our reference MIC is 8. Our AST device shows an MIC of 32, which is resistant. So we do not have essential agreement, nor do we have category agreement. This is also defined as an example of a major error because we have false resistance with our AST device. The last example, isolate 5, we have a reference MIC of 32, which is resistant. The AST test device is showing an MIC of 8, which is susceptible. There is not essential agreement. there is not category agreement. This would be an example of a very major error because the AST device is showing false susceptibility when comparing it to the reference method. Continuing with this same hypothetical example, we have listed here how to calculate the very major, major, and minor errors. For the E. coli, there were 100 isolates tested. 60 of them were susceptible, 30 of them were resistant. We've shown here how to calculate the very major, major, and minor errors. For E. coli, there was one very major error divided by the 30 resistant isolates. That makes 3.3%. Major errors, there were 2 divided by 60. The total number of susceptible isolates, that's 3.3%. For minor errors, there were 3 divided by the total number of organisms, 100, you get 3%. We have the same thing shown here with Citrobacter fruendii also. With the same hypothetical example, we have shown here how to calculate the essential agreement and the category agreement. For Citrobacter fruendii, I want to bring your attention to the fact that because there are 12 total errors, we have a category agreement of 87.2%. The FDA criteria is that it needs to be at least 90% or greater. So if this was the data that a manufacturer found, this performance is unacceptable, and they would have to add this limitation for Citrobacter fruendii versus this antibiotic to their package insert. Next, we want to discuss the verification of an AST system that is FDA cleared. As required by CLIA, a laboratory has to verify that it can obtain the performance specifications comparable to those manufacturers before they perform patient testing with a commercial test system. Laboratories aren't required to repeat all of the testing that a manufacturer has performed. The design on how to design your verification protocol is up to the laboratory with approval from the laboratory direction. We have listed here the summary of recommendations for meeting the CLIA required performance specifications for the verification studies of AST. This chart's listed in the M52 document on page 10 on table 1. For AST, similar to the microbial identification system, you've got comprehensive verification versus a limited verification. We have listed here the types of changes that you would see that would require a comprehensive verification. For the accuracy, we're recommending in this document 30 isolates. Now this should be 30 results per agent. For reproducibility, you would be testing 5 isolates 3 times. If it's a limited verification, we have examples of what would be considered a limited verification. You would want to test a minimum of 10 isolates. Your routine QC, either your 20/30-day plan or your 3x5 plan, would satisfy the reproducibility requirements. If you just have a breakpoint change, we would want to do a minimum of 30 isolates and do your routine QC strains for 5 days. At this point, I'm going to turn it over to Janet, and she's going to be talking about performing susceptibility verification.
Okay, thank you, Beth. And now I'm going to be talking about actually performing a verification of one of the AST susceptibility test systems that we have in our laboratory. As we all know, there are a variety of systems that a laboratory might use for susceptibility testing, and in terms of the verification process, it would be very similar for all of these. Okay, so we're going to go through some examples, and I'm calling these verification exercises. We'll look at 3 different examples. First, we're going to look at verification of FDA-cleared antimicrobial susceptibility test systems when you introduce a brand new system into your laboratory, so you get a new instrument in your laboratory. We're going to talk about how to do the verification when you add a new drug to your system that was previously verified. We're also going to look at doing a verification as a modification of an FDA-cleared test. We're going to use the example here of if you were to update the carbapenem or cephalosporin breakpoints on your commercial susceptibility test system that is not FDA cleared for those new breakpoints. Okay, so let's start with the example where you have acquired a new FDA cleared antimicrobial susceptibility test system. And the question here is, what should you do to make certain this new system will produce accurate and reproducible results in your laboratory? And I think it's very, very important to remember, and as we heard from Beth, when the manufacturer submits their product to the FDA to get FDA clearance of that device, they perform extensive testing. So now it's your job in the laboratory to show that you can attain acceptable performance in terms of accuracy and reproducibility with your instrument, with your staff, and your isolates. So the system has already been looked at extensively by the manufacturer. You now have to make sure it works in your laboratory. So how are we going to do this? And this is just a flow diagram of how we can look at the two parameters that we have to analyze, those being accuracy and reproducibility. So for accuracy, we're going to compare the results with a new system to compare with some reference results. And I'll explain to you in a few minutes how we can get those reference results. We also want to look very carefully at the manufacturer's information because there may be some tips in that package insert that talk about some of the limitations of the system. We heard Beth talk a little bit about limitations previously. And if this is a system that's been out for some time, it's also imperative for us to look at the literature to see if any others have reported any concerns with this new system you're going to introduce to your laboratory. So this can give you some insight in how to develop your verification system to make sure any limitations, any problems with that system can be analyzed during your verification. And then we're going to do some in-house studies. And in the M52 guideline, we suggest a minimum of 30 isolates per panel type to get a minimum of 30 results for every drug you're going to be verifying. For reproducibility, we're going to talk about running the ATCQC strains with each run. We can use those data to assess run-to-run reproducibility. And then in the M52, we say at minimum for reproducibility, you want to look at 5 organisms tested in triplicate. So now I think the other thing we want to talk about is the fact that it's very, very important that you're going to have to perform the testing exactly as described in the manufacturer's instructions. So you really want to look at that package insert very carefully and make sure you're compliant with all those recommendations. The other thing too is that we're going to talk about some breakpoint changes, and it's very important to know what software is on your instrument and what breakpoints are actually FDA cleared in that system. Okay, so we're going to talk now about what you want to do to design the verification study. And it's very important for you to write that protocol before you actually begin performing the verification study. We are providing for you as part of this webinar some supplemental information and actually an example of a verification report that you can use as a guideline for your actually structuring your verification report. So in that protocol, we want to define the numbers of isolates we're going to be testing in each type of isolate, what we're going to consider acceptable category agreement, essential agreement, acceptable rates for very major or minor errors. And we have suggestions for these in M52, but again, too, the final decision on what is acceptable will be made at the discretion of the laboratory director. It's also very important, and we have this highlighted in the M52 document, that it's important to look at those erroneous results that are most likely to do or possibly can do harm to the patient. So we're going to look at risks associated with various types of errors that might be generated from our test system. For example, if we fail to detect an MRSA, this can have some significant consequences. However, if we overcall ampicillin resistance in an E. coli, this may not have such dramatic consequences. So again, too, we have to look at the risks associated with various types of errors. We also have to describe in our verification report how we're going to resolve errors, how we're going to resolve discrepancies. Now, I just want to point out 2 items here. One, in the M52 document, we use 2 different terms. We use the term error. When we talk about errors, if you're comparing your new system with a reference method, such as the CLSI diffusion reference method or the CLSI MIC reference method, we use the term discrepancy if we're using as our method, comparative method, a previously verified commercial FDA cleared method. Okay, so error versus discrepancy depends on what method we're using for the reference results. The other thing, it's very, very imperative that we have to thoroughly train our staff before commencing the verification study. They have to know what they're doing, because some of the results may be as a result of technical errors just because our staff isn't thoroughly trained in the new test system. We want to make sure we analyze each drug separately, and then we're going to calculate the total results if we're doing this comprehensive verification of a new test system. And it's very important to test our ATCC quality control strains each day of testing. Again, those strains that are recommended for inclusion with the panel type that we're analyzing. Okay, so where are we going to get these reference results? So there are basically three ways. We can test our isolates with a commercial FDA cleared method that was previously verified. So if we're going to a whole new instrument, but we have another instrument or laboratory that we've been using for some time, that instrument was previously verified, you could use that as your comparator method. You could also test the isolates with the CLSI reference method as your comparative method, and you could use the broth microdilution or agar dilution MIC test or the reference disk diffusion method as your comparator. The third way is that we can get a set of isolates with known susceptibility results that have been obtained from a verified susceptibility test system or a CLSI reference method. And we can get these from an outside source, and I'll talk to you a little bit later about what some of these outside sources might be. The other thing we might do is we can send some of our isolates to another lab that uses a verified test system. We could send these before we initiate our verification study and then include them with our new test system, or after we've tested them on our new test system to see if that other laboratory gets the same results that we did, and then analyze those results accordingly. Okay, so what criteria are going to be used to determine if the new AST system performance is acceptable? And basically, this is very similar, and I'm not going to go through the calculations because Beth described this. This is very similar to the criteria that are evaluated when a commercial manufacturer submits their data to the FDA for FDA clearance. So we want to attain essential and category agreement that's greater than or equal to 90%. For errors or discrepancies, very major, less than 3%, major, less than 3%. And again, for minor errors, there is no acceptable limit specified, because this is basically included when you do your essential or category agreement. When you're analyzing for very major errors, you want to make sure that only isolates resistant by the reference method are included and calculated that very major error rate. And similarly, for the major error rate, we're only going to include isolates that were susceptible by the reference method. So this is very similar to what we heard Beth describe for the FDA submission data. Okay, the other thing we need to keep in mind is sometimes our new system might perform better than the reference method. So again, here too, we're going to define in our resolution of discrepancies and actually decide how we're going to approach this if indeed we get a different answer from the new system compared to the reference, and which one we're going to consider as the correct results. And all of this is described in our CLSI M52 document. We've included on many of the slides that you've seen the page number where you can actually locate this information. Okay, so selecting isolates for our new AST system verification, as mentioned previously, it's going to be a minimum of 30 isolates for each panel type, because we want to get at least a minimum of 30 results for each drug that we're verifying. And we should say more if possible. And I think if you're verifying a new AST system, particularly one that hasn't been out there for a long time, and you're very concerned about its accuracy and reproducibility, you probably want to include more isolates. Now, in the M52 document, we actually have a table, table C1, that has a listing of the recommended types of isolates you might want to include. So, for example, with your gram-positive panel, you want to make sure you include some MRSA, some DRE, and so forth. With your gram-negative panel, you want to make sure you include some carbapenem-resistant organisms and so forth. And again, this is dependent on the types of drugs you're going to be included on your panel as well. Where can we get these isolates? We can get them in-house from our fresh or stock clinical isolates. We can get them from other laboratories, again, that might have verified the results on their susceptibility test system, and we could either test them again in our laboratory or use the results from that other laboratory. Some AST system manufacturers actually provide a set of isolates you can use for your verification. You would have to contact them about this information. We could use old proficiency testing isolates because a lot of them come with susceptibility results, and you could include those as part of your set of organisms to verify. And then we're going to talk a little bit more about some of the organism banks that are now becoming available. In terms of selection criteria for deciding which isolates to include in your verification, we want to make sure we include representative clinical isolates, those types of isolates we might see in our laboratory, and also isolates that have a variety of susceptibility test profiles. Okay, now we're going to talk a little bit about how we're going to look at reproducibility, and all of this is well defined in the M52 document, where we want to say to test a minimum of 5 isolates, and you could look at reproducibility of your ATCC quality control strains and or clinical isolates, and you want to make sure you are testing those that represent the drugs you're going to be testing on your new susceptibility test system. Test some isolates with on-scale endpoints if these are available, and why I say if available, some of the new very active drugs, it's very hard to find some resistant isolates. Test these 3 times using 3 separate inocula. You could test all on one day or on individual days. And there's really no need to test the isolates with the reference method unless you see failure of reproducibility with your new test system. You can use this for troubleshooting. What is acceptable in terms of reproducibility? We want 95% of the results for an isolate to be within plus or minus 1 2-fold dilution, and this is basically essential agreement of those isolates you're examining for reproducibility. And for the ATCC strains, again, I want to emphasize you're going to include those with each verification run. And 95% of those, so for example, if you do 20 runs, 19 out of 20 must be within plus or minus 1 dilution, and all of these should be, and 95% should be with the acceptable quality control range as defined in our CLSI M100 tables. Okay, now I want to show you an example of how we can assess our results for reproducibility when testing these isolates on our new test system. I'm using as an example cefepime. And you can see that this laboratory tested 5 isolates, and among those were the 2 standard quality control strains. Now, if you look at strain number 1, the E. coli 25922, the acceptable range as described in our M100 document for cefepime and E. coli is 0.015 to 0.12 micrograms per mil. And with the new test system, all of these results were less than or equal to 0.5. which this is the lowest dilution on that new test panel. So looking at these results, these are off-scale results. They're really not a good assessment. This organism does really not well assess reproducibility because the results are all off-scale. Similarly, with the pseudoriginosa, although the acceptable range is a little bit higher, you could see that one of the results in the triplicates is off-scale. But you can see the isolates 3, 4, and 5 that were included for the verification for reproducibility. The E. cloacia is a resistant strain, so this is assessing reproducibility at the upper end, and then Citrobacter frundii and the Klebsiella pneumonia strain all have on-scale endpoints. So these are the types of organisms that are optimal in looking at reproducibility. However, in some cases, it may not be possible. So again, too, when you're designing your study, you might want to include in there to include at least 2 or 3 out of the 5 isolates that have on-scale endpoints. Now, in some laboratories will actually include more than 5 isolates in their reproducibility studies. And again, too, it depends which drug you're assaying, how new your test system, all these other considerations when doing your verification assessment. Okay, on this slide, now we're going to start looking at 2 different scenarios and what you can do based on the results you're getting from your verification study. So the scenario here is to have your new susceptibility test system, and these are the results you've obtained from your 30 isolates of Enterobacteriaceae tested against gentamicin. So in this particular analysis, we included 30 isolates, 10 of those were resistant to gentamicin, 18 susceptible, 2 intermediate. Okay, and as you can see, essential agreement was only 87%. We previously mentioned, and I'm showing on this slide here, the acceptable essential agreement is greater than or equal to 90%. Categoric agreement is fine. So if we look closer at the essential agreement, we can see that this was due in part to 2 out of 10 very major errors. Now here we had two errors of false susceptible results out of 10 resistant isolates tested, giving us a very major error rate of 20%. So we see major, no major errors, and 1 out of 30, the total number of isolates tested, minor errors. Okay, so let's look at these isolates closer that contributed to the very major errors, and happened to be isolate number 5 and isolate number 8. Both of these happen to be Proteus mirabilis. With both of these, the new test system gave an MIC of 4 that was susceptible, where the reference MIC with 16 would be interpreted as resistant. So here we do not see essential agreement, we do not see category agreement, and this is a very major error. So because these were both the same species, and if we got these isolates from our laboratories, we have to ask ourselves the question, could these have been clonal isolates? If they might have been obtained from the same ward, even if they were from different patients, and again, too, we should select isolates from different patients, these might actually be the same strain. We also want to look at any peculiar growth characteristics of this organism that might have contributed to these very major errors or the problems with the results. So this is the initial analysis. And so what are we going to do now here? How are we going to resolve these discrepancies? And we have some guidance for this in M52, and this is one strategy. There may be other ways that you may decide, or your laboratory director may decide, on how you're going to resolve discrepancies. So what we have here, we're saying that we're going to repeat it with both the new and the reference test systems in triplicate. If there are no very majors, we're going to stop. If any very major persists, the next point would be to consider arbitrating these results with the third method, and this might include sending the isolates to a reference laboratory that can use the CLSI MIC reference broth microdilution method. We also might keep in mind that the disk diffusion is a reference method, so you can arbitrate with the disk diffusion method. So nevertheless, with this repeat testing, if your results agree, these repeat results agree with the new method results, we can stop. However, if very major errors persist, we have to consider this drug-bug combination not verified, and we cannot report it on patient isolates. Okay, so here's just an example of the results you might obtain when testing these two problematic isolates in triplicate. And as you can see, when retesting with the new system, the results were basically the same. All of the results were 4 in the susceptible range. But when we retested with the reference method, and again, too, this might have been your old system in your laboratory, now we're getting some results that are different from the initial results that we have observed. So now the isolate number 5 is giving 2 intermediate and 1 susceptible results. Isolate number 8 is giving all intermediate results. And so all of the results now are within essential agreement, within plus or minus 1 two-fold dilution. And although one of them is within categoric agreement, both being susceptible, and that's the third replicate of isolate number 5, the others are not in categoric agreement, but these are all minor errors. So what I'm trying to show here too, and this particularly might be observed when you have included isolates that have MICs right around the breakpoint, that it wouldn't be uncommon to get these types of discrepancies when looking at these isolates. So again, too, when you're describing your protocol for how you're going to resolve discrepancies, this is some issue you might want to address. Okay, so now this just shows the final results for gentamicin and Enterobacteriaceae following repeat testing as part of this verification study. And as you can see, with the 30 isolates, we now have essential agreement of 91%. The other thing I want to point out is because the reference results actually changed for 2 of the isolates. Previously, we had 10 isolates that were resistant. Now we have 8, because 2 of those did not reproduce as being resistant by the reference method. So now we have 91% essential agreement, 90% categoric agreement, and these are both acceptable. No very major or major errors. We now have 3 minor errors, and this is within the acceptable criteria, and so now these results are verified. Okay, so let's look at another scenario, and this is for the test for inducible clindamycin resistance. And this test is on many of the AST system panels. We know this test is essential for those isolates that have the erythro-resistant clindamycin susceptible phenotype, because on these isolates we could not report clindamycin susceptible without looking at the test for inducible clindamycin resistance. So among the staph aureus tested, there were 10 of these that had that phenotype. 4 were susceptible by the ICR test, and 6 were resistant. Okay, when we did the initial analysis, there was only 70% categoric agreement. Also, 3 out of the 6 isolates that had inducible clindamycin resistance were susceptible or were negative by the ICR test. So this would result in a very major error rate of 50%, which is not acceptable. Okay, so what are we going to do here? Okay, so we want to look at the isolates that were problematic, and I'm showing you those on the next slide. And here, this is what we're going to do for discrepancy resolution. Okay, so among the isolates that were examined, that we should be included if we're looking at the ICR test at least one time or several times, we should look at the supplemental quality control strains for the ICR test as listed in our M100 document. So here, throughout the runs, looking at the patient isolates, there were no problems with the quality control strains. And this would not be uncommon. We know those quality control strains perform very reliably. However, isolates number 1, 4, and number 9 were problematic. And when we repeated these in triplicate, isolate number 1 and number 2 still was problematic, being that even though these were positive with the reference method, positive for inducible clindamycin resistance, they were negative by the new method. So this would be a false susceptible result and a very problematic result. Okay, isolate number 9 was repeated 3 times. One of the results came out acceptably, but 2 of the results were still false susceptibility. We decided to add 2 additional isolates to the analysis, 2 more isolates that had inducible clindamycin resistance, and one of them performed acceptably, isolate number 12, but isolate number 11 was problematic. So here, our analysis of discrepancies were to include looking at the QC strains, repeating the three discrepant isolates in triplicate, and then we added 2 additional ICR positive strains. Now let's look at the final analysis where we examined a total of 12 isolates of Staph aureus that were erythro-resistant and clindamycin susceptible by the reference method. Of the 8 isolates that had inducible clindamycin resistance by the reference method, only 4 were detected with the new system. So this gives us a very major error rate of 50%. And now we only have a categoric agreement of 67%. So the conclusion for all of this is that the inducible clindamycin resistant test on the new system did not consistently detect inducible clindamycin resistant positive isolates. This test does not perform acceptably on the new system, so we have to use an alternative test method. So what this laboratory decided to do was to perform the D-zone test on all Staph aureus routinely tested on the purity plates that were inoculated during the setup of the MIC test. The laboratory is also going to discuss this problem with the manufacturer of the new test system. Limited verification when we add a new drug. Okay, so in our M52 document, we talk about those situations where we might want to do a limited verification when we add a new drug, when the manufacturer reformulates an agent, or if we're looking at adding a second instrument to our laboratory. The minimum number of isolates to test for accuracy would be 10, reproducibility to look at QC, either 20-day QC or the 3-by-5-day QC, and this is again the same type of QC we would do for converting from daily to weekly quality control. If there's a breakpoint change, we have to test a minimum of 30 isolates. We want to do QC for 5 days, test those QC strains at least one time each day. Okay, so now let's talk a little bit more about some of the complications that you might encounter when you're adding a new drug to a verified test system. And this is particularly problematic if we're talking about a very recently approved drug. There might be limited or no availability of an FDA-cleared test. There might be difficulties in finding isolates with reference results. In some cases, for some of these new drugs, there are only susceptible strains because resistance is very rare or has never been reported. So this is not essential to include resistant strains in your verification if there are none. And these would be the cases where your diagnostic manufacturer would probably have a note in the package insert that the ability of the susceptibility test system to detect resistance with a particular drug is unknown because resistant strains were not available at the time of comparative testing. So you have to understand what this means, and knowing this information can also help you decide how to approach verification of this new drug in your new test system. Okay, and I want to talk a little bit about some recently approved drugs that might be difficult to verify, and I know laboratories are having problems with this. And drugs you might be asked to test in your laboratory include these new beta-lactamase inhibitor combination agents, ceftazidime, avibactam, or ceftolozane tazobactam. There's only a minimal FDA-cleared test for these drugs. There's disc diffusions by Hardy. Trek Sensitizer does have them on our panels, and there are RUO e-tests available, and also some referral laboratories might be willing to test these for you. And I'm not going to go into the details of RUO testing, but I think we know the RUO test should not be used for diagnostic testing of clinical isolates. I've just listed on this slide here some options where you might be able to obtain reference results from some referral laboratories, and I've indicated laboratory specialists and their phone number because they do have some resources to help with testing these drugs, in some cases for clinical purposes or verification purposes. Also, Mayo Medical Laboratories, Quest Diagnostics, some other referral laboratories might be able to help out as well. Unfortunately, we cannot give you an absolute strategy for how to best approach this in your laboratory, since there is no perfect option at this time. Just a word about verification of e-tests. We're going to follow the rules as for other AST systems. I think when you're using the e-test, it's important to know their FDA clearance status, and this can be found in the e-test application guide, and I've included, if you Google the e-test application guide, you can find this. The other thing too is that I know there are some RUO e-tests out there. Again, these should not be used for diagnostic testing purposes. The other thing I want to point out is that when those RUO e-tests are developed, those might not be the actual type of strip that may be ultimately FDA cleared, because the manufacturer may modify a little bit to get better performance when they're doing the analysis of the e-test strips for FDA clearance and subsequently submit data to the FDA. So you have to be careful about that. Okay, a recent development has been the introduction of the FDA CDC antibiotic resistant isolate bank, and I've included the website for where you can find this, or you can Google the FDA CDC AR isolate bank. And what this is a bank of isolates that have been developed to help diagnostic manufacturers or pharmaceutical manufacturers look at the activity of newer drugs or newer tests on more resistant bacteria. Some of the isolates have contemporary resistant issues. And here's just a listing of all the panels of isolates that are currently available. Now, the intent of this is primarily for diagnostic manufacturers and pharmaceutical companies, but I did talk with CDC, and they said that clinical laboratories can use these isolates for their verifications as well. In my mind, the problem is that some of these are so extensive that might be difficult for laboratories to decide which of these might be appropriate for inclusion in their verification studies. And at this point in time, you cannot just order 1 or 2 isolates from each of these groups of resistant organisms. So for example, if you were to order the panel of isolates that can be used for verifying your carbapenemase detection panels, so if you have a new test for carbapenemase production, there are about 80 isolates in this set, and I know a clinical laboratory is not going to want to test all isolates, so I don't think this would be very practical to get that entire set in your laboratory. Again, this is something you have to decide. The other thing is that I was told by the CDC that even though there are reference results available on all of these organisms, and I show you this on the next slide, that for each of the organisms there is a profile of susceptibility results. However, the CDC recommends that you should not use these results. You should actually retest it in your laboratory by the reference method. So in addition to a nice sheet showing the profile, you could also download an Excel spreadsheet for all of the isolates in a particular panel. So I think as we go forward, there will be some changes made, or not changes made, but some perhaps more information available to clinical laboratories for perhaps how this antibiotic resistant data bank can be used by clinical laboratories to help them in verification studies or to explore some concerns they might have related to resistance in clinical isolates in their laboratories. Also want to mention the ATCC does have a multi-drug resistant reference materials available. I'm not going to belabor this. The one thing I want to mention is that if you want a particular type of isolate, you might find it here, but these are very costly. You know, I saw some of these that cost about $250 in isolates. This is in contrast to the CDC isolates that are available no charge. Our tax dollars are supporting that. Okay, now I'm going to talk about updating the breakpoints on an FDA-cleared AST system that is not FDA cleared for these revised or updated breakpoints. The other thing I want to mention is now manufacturers are starting to update these breakpoints, and even when those breakpoints are updated, you're going to have to do a verification in that circumstance as well, because it is a modification of the system that you initially verified. Okay, so in terms of implementing the revised cephalosporin and carb breakpoints, we now know that most of the revised CLSI breakpoints have been revised by the FDA, so they coincide. Some commercial systems received FDA clearance for one or more of the revised breakpoints, and it's important for you to check with the manufacturer to see what those are. For commercial systems that are not yet revised, we can verify those breakpoints on their commercial system and interpret the MICs with the revised breakpoints. The manufacturer cannot come in and do this for us, but they can tell us how to proceed. So the revised breakpoints, just as a reminder, we're hoping more and more laboratories will use the revised breakpoints, particularly the revised carbapenem breakpoints. We know that they more accurately inform therapeutic decisions, and they eliminate the need to perform some of the special phenotypic tests, for example, for the ESBLs or for carbapenemases, prior to reporting results for the Enterobacteriaceae. Okay, on the next slide, I'm not going to belabor this. This is just a listing of where the manufacturers stand in terms of updating the breakpoints, and also those that are now both updated by the CLSI and the FDA. So where there's a yes in the little box, that means the new breakpoints are FDA cleared on that test system. But again, they might be FDA cleared, but they might not have been yet implemented in the software for that respective system. Also, for the what low dilutions means here is that the panel has low dilutions that will cover the low end of the revised breakpoints, but the system's not FDA cleared with the new breakpoints, so these are ones you can verify because they have sufficient concentrations for you to adopt the newer breakpoints. The other thing too is that the pinkish color here means that the FDA breakpoints do not yet agree with the CLSI breakpoints, so the manufacturer cannot update their systems in those situations. Okay, so verification of the revised CLSI breakpoints. An example, meropenem with the Enterobacteriaceae. We're going to test 30 isolates. We're going to compare only category results because the commercial manufacturer has already obtained essential agreement, so MIC agreement, during their FDA clearance of the drug with their initial submission to the FDA. So we'll only look at changes in interpretation, no changes in the performance of the system in terms of generating MICs. We're going to calculate category agreement. That must be greater than or equal to 90%. Very major errors, there should be none allowed in this circumstance. There could be one major error or one false resistant error. So here's an example of the meropenem old and revised breakpoints. So here's an example of what your analysis may look like when testing 30 isolates to include a variety of species of Enterobacteriaceae and a variety of results for the carbapenems, including some isolates with confirmed carbapenemase genes. So among the 10 E. coli, which included 10 that were KPC producers, all performed fine with no errors, as did the 10 isolates in the other Enterobacteriaceae group. However, if you look at the 10 Klebsiella pneumoniae, one of the 5 carbapenem susceptible isolates gave a false resistant result for a major error rate of 20% among the Klebsiella. Now, if you look at all 30 isolates tested, the essential agreement and the categoric agreement were greater than 90%. But again, among the 30 isolates, which included now 21 susceptible strains, one of the 21 was false resistant for an overall major error rate of 4.8%. And when you wrote your verification protocol, you had to define if one major error rate among 30 isolates tested would be acceptable. And we suggest in M52, when doing this type of verification study, that one major error rate could be acceptable, and this is at the discretion of the laboratory director. So the path forward here depends what you included in your verification protocol up front and what you're going to do now. This might include, might consider these results unacceptable. You might want to repeat them to see if this false resistant result would resolve, or maybe even send the isolate to a referral laboratory. But again, your path forward has to be defined when you write your verification protocol and then decide what to do here to resolve any problems that are observed. Okay, finally, I want to tell you about 2 additional resources that are sources for verification of susceptibility test testing: an older Acumatext that's been around for a while, and also more recently a clinical microbiology newsletter article that talks about verification of susceptibility testing method, and they go into even some additional details, most of what is also included in the M52 document. So in summary, I'd just like to highlight some of the important points about your verification studies. It must be done for accreditation purposes, for CLIA, for your CAP, and in many states they have requirements as well. We did highlight here where you can find the verification requirements in the CAP checklist, and that's in the common checklist. The verification study enables us to become more familiar with the test, so we become more comfortable with performing in our laboratory. It helps confirm accuracy and reproducibility in our laboratory in terms of instrument performance. The system that we have in our laboratory is not a lemon, it's performing well. Operator performance to make sure those doing the test perform it precisely according to the manufacturer instructions. Sometimes enables us to identify problems with the commercial system, and these might not always be detected with the QC strains. It might not always have been detected by the manufacturer when they did their FDA submission, and such might be the case like with the ICR test where we show the example. And I've heard and I've seen here some situation where a verification procedure has actually identified some problems that were not previously reported. Does not ensure all susceptibility results on patient isolates will be accurate, and we need other quality measures, as most of you know, to ensure accuracy of susceptibility results on patient results. Okay, so for ensuring quality of susceptibility results, we know there are multiple parameters we have to consider. We have to do our routine testing of the QC strains. Again, method verification of a new test system or adding a new drug is very important. Proficiency testing, looking at those proficiency testing critiques to see what other problems might be encountered when testing certain drug-bug combinations. Proper attention to instrument maintenance, competency of our staff, using relevant testing strategies. Don't test organism against drugs that are inappropriate for that drug, for that organism. Confirm unusual results. We have this highlighted in our M100 document. Supervise or review reported results. Have a reasonable turnaround time to get these results out so physicians can act on them accordingly, and keep current with changes like you're doing today. So, and a lot of these parameters are defined in your IQCP, so we're not going to go into detail here. I think all of you know where you could find more information about AST IQCPs. So with that, I'd like to thank you and just remind you that no susceptibility testing system is perfect, but we all have to constantly think about the impact of those results we're reporting to the patient. How much might this impact a decision? We cannot rationalize that an error is okay, and we cannot become obsessed by inconsequential errors, those that are unlikely to impact patient management that are unavoidable.
And with that, we must conclude today's webinar. PACE credits are available for those participating in the live webinar only. Within the next 2 weeks, your site representative will receive an email containing the PACE evaluation form. In order to receive your PACE credits, please fill out the form and you will receive your certificate via email in about 1 week following that. Your comments and suggestions are very important to us as they help us to provide you with this kind of quality programming. An on-demand version of this webinar will also be included in the email to your site representative, along with the supplemental documents Janet and Beth spoke about today. To purchase an archived version of any of our webinars, please visit www.clsi.org and click on the CLSIU tab. A special thank you to our presenters, Janet and Beth. If you have any additional questions or comments, you can send them to marketing@clsi.org, and responses will be sent in the follow-up email. Thank you for your participation in today's webinar. We hope to see you here again soon. Today's program is copyright 2016 by the Clinical and Laboratory Standards Institute with all rights reserved. This concludes today's program. Thank you. You may now disconnect.